going to be talking about basically how you make a red flower. This is the work of my postdoc, Julianne Ng. Here's her picture. I realize that's a snake and not a flower. She's a herpetologist, and I hold it against her. So, so this is her postdoctoral work. So a lot of the research in my lab has to do with understanding the origin of different flower colors. We're interested in how they evolve. And from an evolutionary perspective, we're really interested in convergence. And many of you probably know that flower color is a very labile trait. Almost every one of these colors, except maybe teal, perhaps has arisen multiple times on the phylogeny. And the color that we've been focusing on actually are red flowers, which have convergently evolved in many groups, although they're not actually a terribly common flower color. Okay, so the group that we work on is the tomato family, Solanaceae, and I know these don't look like tomato flowers. There are so many cool flowers in the family. So these are 3 of the genera that have red flowers. Out of about 2,700, 2,800 species in the family, there are only 30 that have been described as having red flowers, so it's on the order of 1% of the species in the family. And like I said, this is on par actually with other families. So the first thing that Julianne did was to build a big phylogeny that was enriched for the red species. Sort of ironically, when we started this thing, we realized that a lot of these red taxa are kind of weird little endemics, and they actually weren't in the family tree. So she built this mega phylogeny that has about half of the family, and it's enriched for all of the red-flowered species and their close relatives. So what we were trying to do was get as close to them as possible and figure out how many independent origins there were by sort of filling in those genera. So as you can see, there are a lot of origins of this trait, actually, with 30-some species. Most of those represent independent origins of red flowers. You may notice this funny pattern where there's no red flowers in the top half. For people that know the family, that's Solanum. So half of the family is Solanum. There are no red flowers in Solanum. This other half is where all the other interesting flowers are. Lots of different flower shapes and lots of flower colors on this side. And if you zoom in, what's really interesting, is you see these independent origins, and they're very tippy. So this is among the tippiest traits I've ever seen. I think we just have one case where there are 2 sister taxa that are red. So another way to look at that is if you plot a graph of the depth of each node against the probability, the posterior probability that it's red flower, you basically see here's most of the depth of the tree where there are no ancestral nodes reconstructed as having red flowers, and here are those red flowers tips right under the recent parts of the tree. Okay, so now we wanted to know, okay, so we've got all these independent origins of this thing. How many different ways are there to make a red flower? How many different biochemical pathways are used to make red flowers, which will give us a hint as to how many different developmental pathways and how many different genetic changes might be involved. So ultimately we're asking about convergence here. And so you can imagine sort of 2 alternative scenarios. We look at these different red flowered species, and we find that underneath it's always the same kinds of pigments that are involved. Alternatively, we might find that you can make the same kind of red flower with different classes of pigments. So we know a little bit about this already. There's 3 main classes of pigments that are found in flowers and that contribute to their color. I bet you guys have heard of many of these. The anthocyanins, these guys, these are the things that make blueberries blue and strawberries red. The carotenoids, carrots, bell peppers, but also found in flowers. And the betalain pigments, shown here in some cacti and other caryophyllales. So as you can see, what's cool about these different classes of pigments is that you can make apparently similar looking flower colors with different kinds of pigments, hinting that it should be possible to find multiple pathways in Solanaceae. So I want to mention that in the family, we know that betalains are out. Betalains are only in the caryophyllales and its relatives. So in this family, the only candidate pigments we could find would be anthocyanins and carotenoids. I want you to notice something about their structure here on this slide. Anthocyanins are these small hydrophilic molecules, which relates to how they're stored, and carotenoids are these long, gnarly, hydrophobic compounds. So that chemical difference relates to where you find them in the cell. So anthocyanins are found in the vacuole. Anthocyanins are a pathway that evolved in plants and that and that get tagged with sugars and shunted to the vacuole where they're stored. And so when you look at a cell that has anthocyanins, you'll see that the entire vacuole is colored, so uniformly colored. That's why they call plant cells a bag in a box. So in this case, it'll be a colored bag. And then carotenoids actually are that pathway is of bacterial origin. They're only actually many of the genes have been transferred to the nucleus, but originally that pathway came from bacteria, and these products get shunted back there when they're ready to be stored. So these long hydrophobic compounds can only be stored in chromoplasts, special types of plastids, colored plastids specifically. So this is a picture of some cells, so you can relate this to what actual petal cells will look like. I think these things are just glorious to look at. So here's an example of something making anthocyanins only, so you can see that the entire cell is uniformly colored with a pink color. So that vacuole is full of anthocyanin pigments. Here you can see something that's colored with carotenoids. So it just has those color in those particulate compartments. And so those are the chromoplasts there. And then here you can see dual production. So this is a plant that's making both types, both anthocyanins and carotenoids. You notice that some of these you can only see the chromoplast. That's because petal cells are conical and they're really easy to pop when you're making the slide. So anyway, some of these pop, but that's okay because it makes you more confident that you're seeing the chromoplast in there. Okay, so the point is that with fresh petals, we can really easily score what kinds of compounds are there, anthocyanins, carotenoids, or both. So that's what we did. We went to these 37 taxa, or 34 taxa, sorry, and we scored as many as we could get fresh flowers for. And what we found was that making red flowers with carotenoids is really rare. Even though this is what makes tomatoes red, so there's really no reason to think why it wouldn't be in flowers. It's actually really rare in flowers. Instead, at least as a sole pigment, instead most species are either using anthocyanins or making both anthocyanins and carotenoids. So the next thing we wanted to know is, okay, well, so these are all things that have been described as red flowers. We know that they're making red flowers in different ways. So are these really the same red? How similar are these? Is there sort of a carotenoid red and a dual production red? So we took reflected spectra for these guys, and I'll just be showing you the wavelength at the midpoint of this spectrum. And so just to have in mind something that's more red, the spectrum is going to be shifted towards the red end of the spectrum, so it'll have a higher lambda mid value. Okay. So what we found was, carotenoids are sort of a different kind of red. They're more of an orangey red. They have a lower lambda mid value. Whereas these other pathways, the anthocyanin-only pathway, or making both types of pigments, can give you a pretty similar looking red flower. So these two can be basically indistinguishable. So we think it's pretty good phenotypic convergence. So now we wanted to know, okay, these two things can look to make the same kind of red flower. If we look at the anthocyanin pigments, are those the same? So maybe it's not convergent entirely between the dual anthocyanins, between which developmental pathway they're using, but maybe these dual production ones are still making the same kinds of anthocyanins. Alternately, we might dig in there and see, well, actually there is even less convergence when we look underneath, and the ones that are making plastids, colored plastids, aren't making the same anthocyanin compounds. So in order to understand this, I need to give you a little bit more background about the anthocyanin pathway. So it has these three main branches. So it makes three main classes of pigments. And so every compound, of course, has its own unique absorbance spectrum. And among these, there's some of these that have absorbance spectra that shift them towards being bluer pigments. So if you just had a tube of it, it would look bluer. And some of them are more red. So we have more ones on the brick red end of the spectrum and some purple. So we might expect then that red species would be making red anthocyanins. Pretty straightforward expectation. Okay, so let's see what's in there. We used HPLC to profile these different anthocyanins. So the previous slide just showed the three classes, but in truth, those classes that have subgroups and subgroups and subgroups, we basically colored them by the blue anthocyanins are in blue, the purple ones are in purple, and the red ones are in red. So what you can tell already is that these red flowered things basically can make all of the different types. So dig around, everybody can make one or more of these things. So there's quite a diversity under there. But amidst that noise, we notice kind of an interesting pattern, which is that the species that are making the red anthocyanins, these red pelargonidin-based anthocyanins, they tend to evolve, thank you, in lineages that don't have plastids. So the lineages that have plastids, they seem to be making mostly these blue and purple dyes, and when those plastids, those colored plastids aren't there, we see the evolution of pelargonidin production. And this association between the presence of chromoplasts and the anthocyanin profile is significant with both phylogenetic and non-phylogenetic analyses. So what this tells us is that yes, these two pathways, if we look inside of that vacuole, they're producing different kinds of anthocyanins. So basically, things that are producing red flowers with anthocyanins only are more likely to do it with red anthocyanins. This kind of makes sense. That's the main pigment in there, and it's making red pigments. When you've got those yellow or orangey chromoplasts in there, they tend to be making purple and blue anthocyanins. So it's kind of a funny combinatorics thing. You've got the orange plastids, you put purple and blue anthocyanins on top of it, you get a reddish flower. So that pattern, however, I'll mention was significant, but it was not perfect. We do have some species that are making red flowers in some other mysterious way. This is an example of one of those. This is Cestrum roseum, which is endemic to the Mexican highlands. It is one of these things that only makes anthocyanins in its petal cells, and all of those are those purple cyanidin-based anthocyanins. So what's going on there? We've known for a long time, actually, that there are other things that can alter flower color. So, for example, anthocyanins are part of this flavonoid pathway, this larger flavonoid pathway. There are some slightly yellowish compounds upstream of these anthocyanins, and those can alter how the anthocyanins look. The environment in which those anthocyanins are existing, the vacuolar pH, so if you guys have ever grown hydrangeas and they're blue in one soil and pink in the other soil, thanks. That's the effect of vacuolar pH on the appearance of the anthocyanins. It's not a difference in the chemistry. And then cellular structure, the shape of that petal cell cone can also have somewhat of an effect. So maybe some of these other factors are explaining the exceptions to this general pattern. So I wanted to return now to the big sort of macroevolutionary picture. So we found red flowers have arisen a ton of times in the family. They've done it via 2 or, well, 3 and maybe a few more mechanisms depending on the interaction of vacuolar pH and those other classes of compounds. So why is it that something that evolves so many times in so many ways, why does it remain so rare? So the obvious way to test this would be to apply busy models for those of you that have done some comparative methods. This is a recently developed comparative method, but it has some problems. So I just wanted to mention what my postdoc is working on currently, which is to develop some new simulation techniques to compare alternate models. So specifically, are red flowers so rare because they pop up all the time and then they tend to go extinct? And so in order to ask this question, we're really asking about what kinds of models can give rise to a tippy distribution of states on the phylogeny. So basically, she's taking different versions of the busy model, estimating parameter values under those models, simulating data, using summary statistics to capture the tippiness, and then asking whether this model can generate the kind of tippy pattern that we see on the phylogeny. And just to give you an example, this is one of those summary statistics. It's actually a ratio of some tree distance metrics, MT and PD. And here are 2 of those models. She's tested the evolutionary dead-end model that red flowers tend to go extinct, and the speciation model. And what she finds is that when you simulate under this dead-end model, you can recover a tippiness metric that is similar to what you observe, whereas you don't if it's just that red states don't speciate as often. So this is what we'll be aiming to talk about next year at evolution, but this posterior predictive stuff is a lot of work in and of itself. So we hope to have more on that into next year. And with that, I'll just thank people in my lab, and I think I have a minute for questions. And my funding source.